welcome to this distinguished lecture series episode, which is not on Thursday, but Birgit and I have tried to put together an interesting program for this semester, and this is already our second speaker this semester. I'm very happy to introduce Peter Maurer, who is currently at the University of Chicago, leading a group on quantum sensing and related things, and he's going to tell you all about that. We know each other for a long time. He reminded me yesterday that he was in a class that I taught at ETH when he was an undergraduate there. That justifies my gray hair a little bit. So the class wasn't bad enough to completely make him kind of repel him from the general field. He finished his undergraduate work at ETH. He's actually Swiss. He comes from Zurich. And then went to Harvard and worked with Michel Lukin. Worked on that's where he started working on color centers in diamond, and in particular, he was interested in developing them as sensors, and developed a temperature sensor. And then I imagined that he wanted to learn a little more about biology, and he joined the group of Steve Chu. Steve Chu is a Nobel Prize winner, one of the people who developed optical cooling in the 80s. But he was one of the also first people from quantum optics to become interested in biophysics and single molecule work already in the early 90s. So Peter went to his lab at Stanford and did a postdoc, and then he took on a faculty position at the University of Chicago, where they have a very interesting strong activity from kind of combining quantum technologies, nanosciences, but also life science research, so very much along the line of type of things that we like. And he happens to be in Europe, and we thought to get him to tell us about his current research. So we're looking forward to it.
Okay. Perfect. Thank you very much, Fahid, for the nice introduction and the invitation. I had an amazing 1.5 days. I've been really impressed by kind of the interdisciplinary approach that Erlangen, the larger community in Erlangen has towards sensing and life sciences. And I think much of this, what I'm going to be talking today, will probably resonate with you. So, as Fahid said, I'm by training a physicist. I consider myself a quantum engineer, but literate enough in biophysics to actually be able to express our own proteins, do purification, and be able to talk the same language with people in the life sciences. And so what I would like to do is, over the next 45 minutes, to give you a flavor of where I think a new direction in sensing is heading towards using quantum systems to allow us to probe the physical properties of biological systems that would be otherwise very difficult or maybe even impossible to access. So as a physicist, I'm really truly impressed by the complexity of biological systems and their inherent heterogeneity. So if you take an organism to understand how a complex organism like a human is working, you need to zoom in onto tissue level, where you find something, I believe, on the order of 200 different type of cells. If you look at different health state and different cell cycles, this will be much, much more. And you really need to understand these individual cells to be able to understand how tissue works. And if you zoom further into 100 by 100 nanometer area, you will find this soup of proteins and RNA molecules and lipids and sugars interacting with each other in a complex fashion. And so maybe to be a little bit more biological, there is a lot of information that you miss if you only do an ensemble average. And a somewhat humoristic statement is, if you look at all human, you will find that the average person has one testicle and one ovary. And that's obviously not a good way to characterize a system. And so below is maybe a little bit more scientific example of proliferation of cancer cells. And if you're able to track individual cells over time, you will see that actually benign versus cancerous cells can have very different division rate, and that can allow you to maybe find new cures. And so I would say single cell technology, there has been, it's not a surprise that therefore a lot of energy and money has gone, and manpower has gone into developing new technologies that can allow us to probe biological systems at the single cell level. One of the very first technology was maybe patch clamp techniques, allowing you to probe how neurons fire, and it's now actually widely used in drug discovery. More recently, single cell sequencing that allows us to map out the RNA profile of individual cells. Super powerful technology. Super resolution microscopy, it's really at the essence here that we can resolve individual cells and their structure and protein expression level to single cell mass spec. These are just a few examples that I think have been really transformational. But if we look at all these examples, many people at least would make the argument that they are relying on classical physics, although it's open to debate whether microscopy is a quantum effect. But what is definitely the case is that in precision measurement, the ability to use quantum systems for a measurement over man-made systems has had enormous advantages and consequences. And a famous example is in timekeeping. So people for hundreds of years were interested in building better clocks, because if you can build a good clock, you can use it for navigation. You can send your merchants fleet around the world and become a prosperous nation. And people have gotten actually really, really good in making mechanical oscillators with kind of precisions of or error rates of 10 to the -7. And that has been kind of impressive. But what really kind of made a huge difference when people realized that quantum systems and performing quantum measurements on atoms could give you many, many orders of magnitudes better precision. And nowadays atomic clocks have a precision of being less than 100 milliseconds off over the life of the universe. So these are some of man's most accurate measurement. And as we all know, GPS relies on this, but also fundamental tests of physics. There are actually experiments that can replace experiments done at CERN using atomic clocks, basically. So what my group at the University of Chicago and other people as well are interested in is asking the questions whether we can take these type of measurements inspired from atomic clocks, these super precise measurements, and translate them to study biological systems at the single cell level, at the single molecule level. And the question that my group is asking are, what are the limits of a quantum sensor in a biological environment? How can we perform these measurements? We certainly don't want to perform measurements in an ultra-high vacuum. And what are particular applications and where are particular benefits in the life sciences? It's not obvious at this point where quantum sensors could really make a difference. And so obviously, we don't want to perform measurements with ultra-cold atoms at the nanokelvin temperature in ultra-high vacuum for biology. That would be maybe somewhat pointless. But what we are interested in is using kind of systems that behave like atoms in a ultra-high vacuum, but in a solid state at room temperature, typically. And so a famous example that made actually that created this field of quantum biosensing are nitrogen vacancy centers. These are a crystallographic defect located in nitrogen, where you have one carbon replaced by a nitrogen and a nearby vacancy. And in its negative charged state, this is actually fluorescent and has a spin triplet ground state. Okay, so here you can see a confocal image. Each of these points is an individual NV center. Under optical excitation, you can scatter a green photon and you get back a photon in the red. And interestingly, the MS equals 0 projection of the triplet scatters more light than the plus and minus 1 state. So that allows us to optically read out the spin state of these nitrogen vacancy centers at room temperature. And people subsequently used this platform to perform nanoscale measurements, spectroscopy. Actually, the sequences are completely inspired by atomic clocks. And they were able to see interference fringes, very similar to atomic clocks, but in this case, not measuring time, but measuring magnetic fields. And the idea behind it is that this transition in the 2.87 gigahertz on the magnetic field will undergo a Siemens splitting. And so if you can measure this precision very precisely using this interference spectroscopy from AMO physics, you can infer something about the local magnetic field. And so key to this kind of quantum sensing application have been large coherence times, long coherence times, so maintaining the quantumness of your sensor, and optical readout, efficient optical readout, but also the ability to operate under ambient conditions or in an environment that is biologically relevant. And so we are going to come back to these two conditions over and over again during this talk. So my group is definitely not the only group interested in this. Actually, nearby there are a couple of very good groups. Jörg Wachtrup, who started actually this field in Stuttgart. And so over the years, people have come up with an array of different applications in the life sciences. And so one idea is if you can measure magnetic fields very precisely, you can look at stray magnetic fields from nuclear spins and electron spins, and you can detect maybe perform spectroscopy at the picoliter sample volume, so that gets into the single cell regime. People are also using now these quantum sensors as ultra-sensitive diagnostic devices for cancer detection and virus detection. There are actually two companies that are starting to commercialize this technology. There are ideas to detect action potential, to map out brain activities. There are multiple companies pursuing that direction. And there are new ideas that I've worked on temperature sensing and also like miniaturized patch clamps.
Can you say something about how you say that they're using magnetic fields for detecting viruses?
Okay. I was not going to talk about that because that's not my work. But what they do is there are two different kind of directions. So in cancer detection, what people do, that's a company called QDTI, came out of Misha's lab. What they use is they use magnetic nanoparticles as labels, as an antibody kind of a sandwich ELISA experiment. And they use the fact that in a biological system, you have a very little magnetic background. And so they can detect down to the single particle level with extremely high SNR. So that's this company. And there is a company coming out of UCL And what they are using is they use nanodiamonds as labels, and then they use the magnetic resonance as a way to kind of modulate the fluorescent signal. And so they can get in a paper flow strip, they can detect individual nanodiamonds actually attack to an individual virus. And so they are using this as cheap diagnostics in countries where maybe PCR would be too difficult to perform. So admittedly, these two technologies, they are kind of what we would consider kind of low tech, but that's, I think, how things work. So we first do something in the lab, and then 10 years later, people create companies and make this into actual products. So let me maybe talk a little bit more about one specific application, and this could be maybe kind of a guiding star through this presentation. And that's nanoscale NMR spectroscopy. NMR spectroscopy is this super powerful technology that allows you to get structural information with atomic precision, basically. But it comes as at a price, and the price that you pay is sensitivity. So nuclear spins have very small gyromagnetic ratios, so you end up typically using these 100 microliter sample volume, often in a not very physiological environment. And so over the last 20 years, started by Dan Ruger and others, there has been a big push towards miniaturizing to the level where you can detect single bacteria, small numbers of molecules, even potentially down to individual molecules. And by now, it's kind of clear the technology that is making the race are quantum sensors, qubit sensors. And if you can perform NMR spectroscopy at the micro and nanoscale, you could think of an array of different applications. And one of them is single-cell metabolomics. So if you have tissue, maybe cancer or neurodegenerative diseases, you'd really be interested in seeing metabolic activities of individual cells and combining with other powerful biological imaging techniques and sensing techniques that could really provide important information. Likewise, if we get more sensitive, we could think about detecting post-translational modification of proteins. So we know that a cell contains about 20,000 genes, so we can have 20,000 different proteins. But if you look at all possible post-translational modification of a protein, you will find that there is about 200,000 to 1 million different forms that proteins can be in. And we really don't have good technology to probe those in living systems. We have mass spec, very powerful, but mass spec is very destructive. So NMR could in principle perform these measurements, and if you can do that at the nano or micro scale, that could really allow insights in signaling pathways that are at the moment very difficult to obtain otherwise. Finally, if you have NMR spectroscopy at the micro scale, you could imagine integrating this into high throughput screening of new pharmaceuticals, of new chemical synthesis for environmental control and so on. So the way how this nanoscale or microscale NMR spectroscopy works is basically you have a molecule that contains nuclear spins. These nuclear spins produce a dipolar field, a magnetic dipolar stray field. And if you can bring a sensor very, very close to this, you can detect actually quite sizable fields. So the dipolar field falls off as 1 over r cubed. If you look at noise correlation, it's actually 1 over r to the 6th power. So getting close with a sensor really buys you a lot. And once you can detect this stray field, you can actually use techniques from established NMR spectroscopy to get structural information out of the resonance spectrum. And really early work on this was pioneered in Stuttgart, Harvard, and UCSB and IBM. So where are people currently standing with this? There has been a lot of effort in this direction. And I would say we are currently, people are pursuing two directions. One is spectral resolution. Multiple groups simultaneously came up with ways to get resolution that is not limited by the coherence time of your quantum sensor. So in principle, whatever is the coherence time of what you want to detect of your molecule, you can actually resolve. And in parallel, people have pushed sensitivity limit. This is work from Misha Lukin's group that I was not involved in. And they actually put ubiquitin proteins on a diamond surface that was labeled with deuterium and carbon-13, and they could see resonances. So when I ever talk to my friends in physical chemistry, they always say, yeah, it's kind of cute experiments, but the spectral resolution is not where we want to be. This, I mean, is like percentage chemical shift. This is still like in the many ppm range. And literally, I talked to established physical chemists here in Europe that got visibly upset with me when I talked about NMR spectroscopy to them. And so the goal is to go to actually high magnetic fields, to go into get into the ppb range. And so when I tell this, and the thing is actually, I can, let me illustrate this example. So this is a small molecule that Ron Walsworth's group actually performed this quantum sensing experiment, and they get spectra that kind of resemble what they would simulate. But the problem is at these low fields, nuclear spins have crosstalks, and you can not do secular approximation, and you end up with very complex spectra that in principle for a more complex molecule you would need basically a quantum computer to simulate the spectrum. However, if you go to large magnetic fields, the chemical shift between different nuclear species is large enough that the spectra start to become very, very simple. And so my friends in physical chemistry, they say if you have 14 Tesla, you really can start doing relevant experiments. The problem is to go to 14 Tesla is actually kind of a challenge. And the problem is our NV centers, we need to perform coherent manipulation on our qubit. And at 14 Tesla, we are talking about 400 gigahertz microwave frequency. That's a very unfortunate energy scale. So in the few gigahertz range, we have off-the-shelf components that are really cheap by now. In the optical regime, we have also off-the-shelf components that are really cheap by now. But in this terahertz gap, the technology is not there. And so I've teamed up with a couple of my friends at the University of Chicago, and we started to explore what would be needed. And I think it's as much of a scientific challenge as an engineering challenge to develop such a system. So we've taken a 14 Tesla NMR magnet, an old Brucker magnet with a room temperature bar. We engineered together with a company, Thomas Kitting, this free space upconverted or this free space amplified and frequency doubled system where we have 400 gigahertz at 50 milliwatt that we can send onto our sample. So the sample is here, it's a microfluidic channel, and we have optical readout of our NV center. But 50 milliwatt is actually not good enough to strong enough drive your NV centers and your quantum sensors. And so my colleague, Alex Hai, kind of came up with the idea to use meta materials. And with this, we can now actually get quite decent Rabi oscillations. And so this is an ODMR spectrum now at 400 gigahertz. So we started to benchmark our quantum sensors. At 14 Tesla, your nuclear Larmor precision frequency is 600 megahertz. That's actually too fast oscillating field that the standard quantum sensing protocols could be applied to. So we came up with a new protocol actually inspired by a group in Spain. and we measured a dummy signal. This is not yet nuclear spins, but a dummy signal, and we can actually get pretty narrow spectra. From this measurement, we can extract what our sensitivity is. It's about 100 nanotesla per square root hertz per micrometer cube. So ideally, we would want to be at the sensitivity in nanotesla. I'm going to talk about this in a second. We think with some simple improvements, we can go to the nanotesla sensitivity. So one of the first experiments that we are envisioning is application in single-cell metabolomics. And the idea here is basically the following. If we can get to the nanotesla range, we can detect molecular concentration inside of a cell in the 100 millimolar range in a second. So if you look at bacteria, that starts to be some of the first metabolites. If we can further improve the sensitivity using established techniques like hyperpolarization, we should be able to go to the millimolar range where really many of metabolites are located. And so this could be an interesting thing to study biofilms. With further improvement, we could think about cancer biology and neurodegenerative diseases. So that has all been kind of following trajectories of pretty established technology on the quantum sensing. This is about 15 years of work in the community. And we are now, I think, at the threshold of getting to measurements that could be really useful for biology. But these measurements are all based on like millimeter scale diamond chiplets, where you put your sample on top. I think it would be really amazing if we could have a nanoscopic quantum sensor that we can bring into a cell, into a living system, to perform quantum measurement, NMR spectroscopy locally. And it turns out that actually you can crush up your bulk diamond into nanodiamonds, and cells can uptake these nanodiamonds. Here I'm showing a fairly old publication from a group in Australia, and each of these red spots is a nanodiamond inside of a HeLa cell. And sensitivity are actually still decent. Coherence times are in the microsecond range. However, the sensitivity in these nanoparticles and coherence times were significantly worse than in bulk diamond. And the reason is that we have now surfaces in these nanomaterials that are kind of difficult to control for. And so this is an TM image of nanodiamonds. And if we measure coherence time, some of these can have actually fairly good coherence time, but other nanodiamonds have pretty bad coherence time. That's not great if you want to use this for a bio experiment. You want to have each of your sensors to be kind of identical. Yeah, so we really were kind of limited by surface defects, and we were thinking about ways to get around this problem. And we looked at people in the nanotechnology community, and when they started to look at quantum dots, they saw kind of this interesting single photon emitters, but quantum dots at the time were really not that useful. They were keep blinking. And people realized that this was actually due to surface charge traps. And the solution was to encapsulate the quantum dot in a protective shell that would result in this stable fluorescent signals. And while this is done for just optical properties, we thought maybe we can do a similar thing for coherence. And we encapsulated our nanoparticles with a silica shell, and we saw a significant increase in coherence time, and actually none of the diamonds that we studied had this very low coherence time. So that's through material engineering. We can now get materials that has improved quantum properties and reproducibility. This paper has actually just a week ago come out in PNAS. So that's all interesting, but what could be some of the applications we could use these nanoparticles for? They are not good enough yet for NMR spectroscopy or EPR spectroscopy. So as a result of that, we've been thinking about maybe some other application. And one of the things that is actually known about diamond is when you shine light on diamond, you have a photo-induced redox reaction on the surface. And that results in a loss of electrons, a buildup of surface charge. And that surface charge actually has, over a second order effect in the spin Hamiltonian, reduces in slight changes in the zero field splitting in this transition. Now, that may not look like much in this measurement, but believe me, with this high precision spectroscopy that we are applying, these are huge changes. And we tested this hypothesis by adding a silica shell that now acts as a tunneling barrier. And we found that this results in a reduction of a charge buildup, and we have now much more stable zero field splitting lines. So we then went a step further. We brought our nanodiamonds into raw cells. Raw cells are a type of model system to study macrophages. And what we found is for the bare cells that our zero field splitting, our qubit transition, is actually kind of diffusing around. There's quite a bit of diffusion in the transition frequency. If we use our core shell structure particles, that diffusion has reduced quite a bit. And so that's actually a suggestion that there is inside of a cell kind of funny charge dynamics ongoing, maybe related to this redox mechanism that I mentioned before. Now, that would be just kind of an annoying fluke, right? However, macrophages, they can actually be stimulated. And so what macrophages do is they look for bacteria in our immune response and then engulf them and eat them and kill them. And what bacteria do is they actually excrete a particular type of sugars. And so these macrophages, they can find the sugar. The specific one is LPS. And they get kind of activated. They become hungry when they detect this sugar. And so what we found is that if we activate macrophages with LPS, we actually see a consistent change in zero field splitting. And that is not observed in our control experiment where we use this core shell structure. So this is actually kind of, to us, was a surprising observation showing that we can detect kind of cellular processes using these quantum measurements. We kind of did not expect that. We kind of stumbled into this. And we are now working with people in the medical school and in immunoengineering to generalize these measurements and apply it to understanding and T-cell activation in tumor killing. And there's quite a bit on the quantum side that we still need to understand, and we are building instrumentation to do these measurements in wide field experiments. So with that, let me transition to the last topic I wanted to discuss today. And I'm going to trash my previous work a little bit. So the work I've shown so far was all based on diamond. And while there are definitely insights we can gain, nanomaterials are a pain to work with. Nanoparticles are a pain to work with. And the reason for that is that nanodiamonds that have good optical properties tend to be large. They are typically 50 to 100 nanometer in size. Your average protein is on the order of 3 to 10 nanometers. So if this is a protein to scale, you would have a nanodiamond like this size. And so I'm living in Chicago, and we have this sculpture. It's called the bean. And it measures from here to here, from end to end, 20 meters. I'm 2 meters tall. So the size of this bean to me is the same as a nanodiamond to a protein. And nobody in their right mind would make the argument that if you glue me to this bean, you can observe my normal functions. So the same thing is true for nanoparticles. The good thing is if I present here, I can use meters and people don't get confused. So what biologists really like are genetically encodable probes. And so why they like it is because we have really good, we know from genomics where genes are that encode proteins that we want to study. And we have really good molecular techniques, scissor and glue basically, to insert a gene that we want, in this case, a fluorescent label, a green fluorescent protein, into the DNA right after our favorite protein. When the cell, when the RNA polymerase transcribes the information in this in our target gene, it actually also reads the information from the fluorescent protein, and it creates this RNA sequence that now has both information on it. Once the RNA gets translated, we end up with a fusion protein that is kind of the super protein that consists of both our target protein, now covalently linked to the fluorescent label. And I cannot stress enough how important that has been for biologists. There are now data banks out there with thousands of different plasmids already labeled some of the most interesting genes. AdGene is, for example, a database like this. There is the Allen Institute that has large libraries of cells that you can just order. And there's an entire ecosystem built around this technology, including a billion-dollar imaging technology. And so circling back to comparison to nanoparticle, this is still kind of state-of-the-art delivery of nanoparticles into cells. And most of these particles are actually going to be in the endosome. Versus here are microtubules genetically labeled with fluorescent proteins, mice that are transgenic. I stopped using the word transgenic mice in the US because our current administration confused it with transgender and wants to cut funding. This will not be accessible in the US, right, Fahid? The recording. And I visit you permanently. I'm not sure if you want that. The other thing I want to point out, besides labeling, these sensors are actually really, really small. That allows us to go 10 times closer to the target protein. And being 10 times closer, a dipolar field from an electron or nuclear spin will be 1,000 times stronger. Again, if we look at nuclear noise, it will be 1,000,000 times stronger. And so these two parts have been really motivational for us to start looking at fluorescently genetically encodable probes as qubits. And this is actually, maybe in hindsight, I did a risky or a stupid thing that you shouldn't do when you're on tenure track. But I thought, let's start with just a completely new system that may work or may not work. And so we took this fluorescent protein, yellow fluorescent protein, one of the favorite proteins that biologists use. And it has this barrel structure. And in the center of this barrel structure, you have a fluorophore that is consisting of 2 amino acids that get linked together. And when biologists use this for fluorescent imaging, they excite with 488 and they detect a photon in the green. Interestingly, once in a while, you have an intersystem casting into this triplet state, that is metastable. Usually, biologists really hate that triplet state. It reduces fluorescence, it induces phototoxicity. However, it happens once in a while. This intersystem casting is actually spin projection dependent. So by just shining optical light, you actually build up polarization in the triplet state. And it's this triplet state that we are using as a qubit. In principle, you could wait for the triplet state to decay through phosphorescence and detect this phosphorescent photon, but that's horribly slow because this lifetime is in the millisecond range. Actually, one of my students came up with this idea, which is a really good idea. You can excite with a near-infrared laser to higher-lying triplet state where you have faster intersystem casting. It's, again, spin projection dependent. And then you get a fluorescent photon back. And so you can, by shining this near-infrared light, you can detect the fluorescent photon and use that as a readout of your spin state. This process is known as optically activated delayed fluorescence. It's a fluorescent photon because it's a green photon. It's delayed because fluorescence usually comes back in a nanosecond. This takes a microsecond. And it's optically activated because we need this near-infrared laser for excitation. Also, I should point out that while the photon count rate here is low, it's completely background-free. There is no autofluorescent background at the microsecond timescale. And so for those of you familiar with NV center, if we forget about this ground state, this is actually the level structure of an NV center. And I've actually been thinking if we could maybe drive this transition directly to do some readout as well. So now how our experiments work is we initialize with 488, so we pump into the triplet, then we coherently manipulate the triplet state with microwave pulses between TC, TY, and TX, and then we read out through this optically activated delayed fluorescence through this transition. Okay, we can show that we can actually detect optically a spin state. We use an ensemble of our yellow fluorescent protein, and we initialize, apply a microwave pi pulse at varying microwave frequency, and then read out. And so if the microwave frequency hits a resonance between TC and TY or TC and TX, we see a change in OADF signal. And now we can show that this is not a fluke from our instrument. We can apply magnetic fields, and we see distinct shifts in our resonances, which are Zeeman shifts. And we know they are Zeeman shifts because we can actually map out the OADF response as a function of magnetic field, and we can compare it to just the diagonalized Hamiltonian. And the two things agree actually pretty well. Now we can go a step further. So far, this is could be also incoherent spectroscopy, right? We can apply microwave pulse of varying duration, so we can Rabi drive our molecules, and we can see this characteristic Rabi oscillation. Now, admittedly, these are not the most beautiful Rabi. But it's not because of coherence, it's because we measure multiple proteins, an ensemble of proteins. They have all different orientation, and so the Rabi drive frequency is kind of over a broader spectrum. That's why it washes out. Okay, so we can study actually spin coherence. We can apply a Hahn echo and then do our optical readout, and we can get at liquid nitrogen temperature, we can get about a microsecond spin coherence. This is actually kind of long if you think about it, because in a protein you have a lot of protons, and protons have fairly large gyromagnetic ratio, and that typically should dephase your electron spin. And we are kind of actually lucky with our system. We have a relatively large zero field splitting, and what that does, it gives you this, for low magnetic field, a second order sensitivity to perturbation in magnetic field. In atomic clocks, people call that a clock transition, or in precision measurement, people call this a clock transition. And indeed, if we go to a larger magnetic field where we are further away from the second order effect, closer to the linear regime, actually coherence time decreases. We can compare the phasing rate as a function of magnetic field to the black is our model, and the two agree actually pretty well. So now nuclear spins are actually known for being typically a non-Markovian noise environment. And so we can use more advanced decoupling sequences. A famous one is called CPMG, that is a train of pi pulses. If we use 240 pi pulses, we can actually get coherence time on the order of 15 microseconds. And using the observed noise and signal, we can convert this into a sensitivity of 183 femtotesla square root mole per square root hertz. A nanodiamond, an NV center in a nanodiamond, has about a microtesla per square root hertz sensitivity. So far, this has all been at liquid nitrogen temperature. We can now study what happens at room temperature. So we first study depolarization rate of the spin. So that's a transition from TC to TX or TY, unwanted transition. At low temperature, we have actually fairly long T1 times in the millisecond range. At 100 millikelvin, this becomes about 100 microsecond, and it's a linear dependence which comes from a phonon scattering on our spin, inducing a transition. But at higher temperature, this becomes actually T to the 7, temperature to the 7 law, and that's a Raman transition where Tc absorbs a photon to a virtual state and then emits another phonon to T via Tx. And the theory behind this predicts T to the 7, and the experiments actually fit this pretty well. So nevertheless, at room temperature, we still have about 250 nanoseconds depolarization rate. That's enough to observe ODMR, so optically detected magnetic resonance. And we can apply magnetic fields. We observe shifts in the ODMR resonance. We can now measure the shift by measuring 2 points and look at the imbalance of the signal, like what people do in atomic clocks, as a function of a magnetic field. And we can, from this curve, we can extract the sensitivity at room temperature of 93 picotesla square root mole per square root hertz. Okay, then just to drive the point home that these are biocompatible systems, we transfect mammalian cells, hex cells, with a plasmid that contains YFP. Here you can see individual cells. Blue is OADF signal from the cells. Inside of the cells, we see resonances. Outside of the cells, we don't see any resonances. So with that, let me give a 5-minute outlook on where I think potential of this technology lies. So we are still kind of far away from doing single molecule experiment. We detect maybe a femtomole, a few 100 million molecules at this point. But that actually kind of gets into the range where a heavily upregulated protein is in a single cell. And so what I imagine is we have this heterogeneous tissue. Now we have inside of a cell genetically encoded our sensor tagged to the target protein. We have many copies of this, and we use a confocal microscope to read out the single cell. Again, the length scales here are 3 nanometers. And there are 2 things that improve our sensitivity if we want to perform NMR or EPR spectroscopy on it. One is the short distance, and the other one is that we have n copies of it, which gives us the square root of n enhancement. So if I put this on this maybe somewhat provocative plot, if we compare the qubit target distance, so that's this distance, to the sensitivity, the measured sensitivity of our sensor, we are here where this star is. That's for a femtomole, kind of what you could get inside of a single mammalian cell. Now, if we are below this line, we are capable of detecting an electron spin in a second integration time. Below this dashed line, a nuclear spin. People have been for 20 years kind of trying to get into this regime. These are some of the best values measured with other platforms. Squares are technologies that you cannot bring inside of a cell, like the bulk diamond that I showed before, or a squid or a vapor cell. Circles are nanodiamonds. And again, we are with our technology here at this star. If we were to extrapolate our measurement to a single protein, we will be right now up here. Okay? So with this, we are, by the way, already way more sensitive than your best conventional EPR spectrometer. So just to give you a few numbers. So the sensitivities that we demonstrated are on the order of 200 femtotesla square root mole per square root hertz. Single electron spin 5 nanometer away from our sensor, which is a typical first resonant radius, gives you 14 microtesla field. So now you can calculate how long you would need to integrate to detect your volume of or your particle numbers. A single nuclear spin about 20 nanotesla. But I think there are other things that our quantum sensors could be very uniquely positioned to measure that still need to be explored. Because we are so small, we can be within the Debye range of charge states. Maybe this could be interesting to probe chemical environments or ion channels. Now, so far, we've always been, in my grant applications, I was always say, understanding quantum, we can actually develop sensors that can be used in biology, but maybe we should also ask the other question, what can molecular biology do for quantum? And I think there could be some, with this biological qubit platform, there could be some really unique opportunities. And one of the things that I think could be interesting is engineering coherence in our qubit system. So we have our qubit, our protein that is encoded in its blueprint is in the DNA. Now we can mutate the DNA and get many different proteins. We can measure which has an improved optical or quantum property, and then use this DNA as a new starting point in mutation. So this is actually a process that people used for a long time in protein engineering. It's called directed evolution. And nature has used it even longer from originally we were super fun structured molecules, and we are now these complex organisms, and this all relies on this kind of black body optimization. And maybe there could be actually quite interesting insight gained from this on coherence in open, noisy quantum systems. Another direction that I think could be interesting is engineering quantum systems with atomic precision. So people have spent a lot of money to create NV centers with one or multiple nuclear spins nearby and arrays of NV centers, and we are still not able to do this kind of reproducibly with nanoscale accuracy. Yet, biology is really good in that. Look at DNA. You can get periodic structures in 1, 2, 3-dimensional with DNA origamis. You can use isotope labeling to get nuclear spins in specific locations. This could actually be a new opportunity to simulate complex quantum systems that current diamond-based technology has a very hard time to do. And so with that, let me wrap up. I started with this microscopic scaled sensors, these diamond chiplets that are capable of probing single cells. I then went to engineered nanoparticles that are the size of a cell organelle that we are now using to probe the interior of cells. And most recently, we've developed these protein-based qubits that are really on the size of a protein that you want to detect. With this, I'd like to wrap up and give you my point of view where I see quantum sensing standing. I think quantum sensing is kind of where single molecule biophysics was in the early 90s. So in the early 90s, people were able at low temperature to detect the fluorescence of an individual pentacene molecule in p-terphenyl. It took about 15 years of scientific and engineering development to go to super resolution microscopy, single molecule microscopy. And that then didn't take that long anymore for industry to make commercial tools. I think with quantum technology, we are somewhere in between here. There are definitely some first applications starting to be seen, but there's still a lot on the scientific and the engineering front that needs to be addressed.
Talk with lots of different aspects and questions.
For the yellow fluorescence that you are putting in a protein, did you already know the level structure and where these?
Yeah, that's actually why we chose the YFP. I think in hindsight, YFP was a unfortunate bad choice because it doesn't have a large intersystem crossing. That's why biologists like it. But there has been a paper maybe 2 years out that did spectroscopy of these higher lying triplet states and actually for the first time described this OADF readout. They didn't do any spin physics on it, but they showed that you can pump the triplet back into the singlet. They actually wanted to show that this makes the YFP brighter. But having had that information available was for us a good starting point to start with a system where we knew what lasers we had to buy. We are now working on actually sweeping the entire libraries of dozens of fluorescent proteins for systems that work better, and then we will, based on that, do directed evolution. But you know, the interesting thing here is that biologists, when they choose fluorescent proteins, they kind of want exactly the opposite from what we want. They don't want to be in the triplet. So I think there is, we can improve quite a bit, and there will be a lot happening in the next years in this one.
So you're like
Back there. First off, great presentation.
Thanks.
Second off, so it seems like the general theme, this broad theme of this presentation is more about was quantum sensing at very small scales, and like you mentioned, optical clocks are big in that area. Considering you're at the University of Chicago, which I associate nuclear physics. I was just curious. You mentioned optical clocks. Apparently, I think I read an article that by the NIST, there was something called the development of a nuclear clock. I was just wondering if there's any.
Let me just reiterate. So we are not using atomic clocks. We use ideas inspired by atomic clocks. Because the nuclear clock, I think, is really cool. Juni's group had some nice paper, and actually here in Germany, some of the early work was done on this. Nuclear clocks have the advantage that you can load a mole of nuclei to probe, right? So you could get, in principle, really, really particle numbers, and you don't have any kind of thermal, any redshift from thermal motion. So nuclear clocks are amazing. I think this will be another huge step. I'm not entirely sure how this could be useful in our experiment. Maybe nuclear spins can be useful as if we use the fluorescent protein as a quantum register. People have already used that with NV centers, where you have a nuclear spin as a memory and the electron spin to address one or multiple nuclear spins, and you can build actually a small quantum computer with it. It doesn't scale, so that's why people are not using it for quantum computing, but you can make simple algorithm. Back in my PhD, we actually had a paper where we use quantum logics to improve the readout fidelity by 2 orders of magnitudes this way. So maybe you could use now fluorescent protein where you have multiple nuclear spins deterministically positioned, because we know where our amino acids are, and use that to get a quantum advantage in your sensing. Those are definitely things we are thinking about. But again, then the only kind of connection to the nuclear clock would be that both use nuclei. But thanks for that question.
Oh thanks for the answer.
Yes, just on the YFP also. So you essentially only rely on the statistical transition to a triplet state, right? So it's
So it's about, I think the yield is about 1%, something on that order. And that ideally we would want to have 30%. Now,
the variants of the fluorescent proteins are all much worse in that regard. So even
Yeah.
is already improved to have less.
yeah.
And the second question is going to the nitrogen vacancies in raw cell.
Yep.
In raw cells. So this shift upon activation, if it's related to a redox state shift, that would be related most likely to mitochondrial signaling. So did you inhibit that, like mitochondria or something?
No, we haven't done that yet. it's possible. Mitochondria are definitely something we want to check. The other thing is, I'm even not sure if these nanodiamonds are actually released from the endosomes.
It wouldn't matter.
So-
I mean, like if the redox state in the cell changes, then simply by sort of changed gradients between the cytosol and the lysosome, you would still get some effects. So that thing goes like if you starve the cells or induce any sort of actual stress like this will all directly affect mitochondrial signal.
This is a good, we should talk a little bit more because one of the reviewer was saying, can you please do some biological control on this? You formulated it nicer than the reviewer.
I mean, to that extent, I think a little related is fluorescence lifetime imaging. So, you know, sometimes people come up with explanations that you have something, and then if you bring it in this situation and the other situation, lifetime changes. But the fact is that lifetime changes no matter what you do. And I think people use fluorescence lifetime imaging in practice, but more as a kind of a pragmatic way that you look at it and then you make a category of the situation and this whatever, this is how much it changes.
Yeah.
So if you make that, this is probably what it is, but not so much from a first principle that it changed by twice and therefore this was the mechanism. So I think that's where many mechanisms come in. You still becomes useful, but maybe not.
Yeah.
In its kind of pure form.
I think we've really just started understanding these systems, or understanding kind of in quotation marks. My hope is, but that's just my hope, that the spin transitions, we have a 3-level system, so we have a little bit more diagnostics than with a lifetime measurement. And my hope is that if for lifetime there are 10 different things that can change your lifetime, still means it's still biological meaningful if it tells you something. Here maybe we will end up with 5 different or 3 different things. And for the simple reason that we have quite a lot of quantum control techniques to kind of learn what the nature of the noise is that produces these effects. But you're absolutely right. When I talk to my friends in biology, they say, Peter, why do you care what's the physical mechanism? We see a change and that's all we care about. Yeah.
I mean, at the end, I think it's all about information, and whatever you do that gives you more information is useful.
Yeah.
And that's why everything that you've shown is exciting, even if it doesn't work the way you want it. So, if there are other questions, there's reception outside, so you can continue to interact with Peter. There's a question from.
There are 2 questions from the.
All right, let's have a look at them. Here are your questions.
How many atoms has a nanodiamond? Oh yeah. So our nanodiamonds are big. Our nanodiamonds are 50 to 100 nanometers. I think I calculated once, like a 5 nanometer nanodiamond has something on the order of 10,000 carbon atoms. So if we go to 50 nanometer, that goes as the cube, that would be 1,000 times more. Yeah, so something on the order of 10 million atoms. But yeah, I guess you can do that calculation as an exercise as well. Have you considered collective spin squeezing to improve the NVC diamond magnetic field sensing? Yes, we have actually a theory paper in NPJ quantum information, how we could use spin squeezing to improve sensitivity. And squeezing is actually kind of one of these interesting things. It turns out that entanglement by itself does not guarantee you a sensitivity increase, because entangled states are more sensitive to the environment that you want to sense, but they are also more sensitive to the noise. And actually the 2 effects cancel out each other. There are 2 reasons where entanglement can help. that people have known so far. One is if you want to have a high bandwidth of your measurement, where you want to make sure that you can perform your measurement really quickly, even though the sensitivity is the same. The other one is in atomic clocks, if your noise is correlated, then you can actually take advantage of it. I think in these kind of diamond systems, or proteins as well, noise tends to be correlated because these nuclear spins, they are like non-Markovian environment. The other thing is that's the thing. So noise can be correlated, so this could actually give an advantage. So now the question is how to create entanglement or squeezing. And we've shown in our paper that even for an unknown spin Hamiltonian that is dipolar interaction, you can actually use global control and evolution in combination with a learning algorithm to drive your system in a squeezed or an entangled state, in a metrological useful entangled state. It then turns out that if you go close to the Heisenberg limit, that state happens to be actually a squeezed state. However, that's a theory paper. There is quite a bit of work we need to go to actual experiment. But I think some of these spin chains that I was mentioning at the end, using kind of DNA origami, could actually be an interesting way to create squeezing. And I'd be happy to discuss this more afterwards, but the person is not here. Yes. So with NV centers, we can actually get really high readout fidelity. People shown 99.99%. So in principle, you could really be in the shot noise limited regime. Whether in practice, if you add everything together, like multiple NV centers next to each other, if you still can get this 99.99% readout fidelity is another question. Yeah, I think it's at the moment more like a Gedankenexperiment.
All right. Well, please join me in thanking Peter again.